Well, I would like to thank everybody for joining us again for our University of Idaho, Utah State University, and University of Wyoming Sheep and Goat webinar series. Today, I am your host, primarily Melinda Ellison with the University of Idaho Sheep Extension. And on other days, we also have Chad Page from Utah State as the small ruminant specialist, Carmen Wilmore with the University of Idaho as an extension educator, and Whit Stewart from the University of Wyoming, sheep specialist. And so those are the four of us that host this webinar. And so thank you again for joining us today for the live version. As always, you can catch our uploaded versions of any webinar that we've done on our YouTube channel, which is the University of Idaho Extension Livestock channel. With that, I would like to introduce today Dr. Chris Posberg, which is a tongue twister for everybody probably. But he's joining us from Montana State University and is going to discuss with us using genetics to improve profitability in our sheep and goat flocks. So welcome, Chris, and take it away.
Great. Thank you for the introduction, Melinda. I appreciate it. And I'm glad to be here with you guys today. And to get us started, I'll just do a quick background, a little bit more on what I've done and what led me to my position here. So I'm originally from New Jersey. Please don't hold that against me. Grew up on a small sheep farm there, raising mostly Romneys and some Dorsets, which you can see in the bottom right-hand corner there, and mostly 4-H hobby flock, developed mostly for the Romneys for hand spinning fleeces and that niche market, and the Dorsets kind of for market and freezer lambs. But from there, I went and pursued a PhD at Cornell University in animal science, did most of my work on small ruminant genetics and genomics, looked at a number of economically important traits, so aseasonality or out of season breeding, mature body size, and then coat color for those hand spinners, as well as looking at milk production, estimated breeding values in goats down in Australia. And I'll say today, for those of you that are raising goats that are on the call, while I do focus mostly on sheep in the presentation, this can all be applied to goats as well. And then I've actually been at Montana State here for 2 years on the day, where most of my work now is focusing on wool quality, some feed efficiency, and tying genetics and genomics and utilizing some of those tools to implement some of the precision ag technologies that are emerging to try and improve sheep production overall, whether that's wool or lamb production. So to get us started, I'm really going to kind of do some basic background information for those that may not be familiar or just want a refresher. I'll refer to these kind of as we're going through the talk today. So a breeding value, this is just that value of the animal as a genetic parent. So how valuable is that animal in passing on the traits you care about to its offspring? Then we've got genes and alleles. Genes are really any stretch of DNA or RNA if we're dealing with some other species that determine a certain trait. So we have genes that influence coat color, growth rates, things like that. And that's just a stretch of DNA that impacts that. And then we can have different versions or different variations within that DNA sequence. And those are what we call alleles. So it's one of two or more versions of a gene. And really it's just saying that there's a different sequence there or a different string of letters there, but it still is affecting that particular trait. When we talk about phenotype, this is what we physically observe. So this is, we can see that coat color of that animal, or we can collect our weaning weight information and get that weight, and that is that phenotype. Then the genotype is what that DNA says should be observed. So based on that sequence, we can do our, so those individuals in my role, we do research studies to figure out, okay, our genotype says the DNA codes for this, and we should see an effect size of about this size on that particular trait. And when we think about DNA, and should have prefaced too, my background is more so on the molecular side of genetics and genomics, a little less so on quantitative approaches. But when we think of DNA, one good metaphor for it is to treat it as a library. So the library is every information, all the information we can have in a cell. And, as geneticists or sheep breeders, we do have to factor in everything. We have to factor in nutrition, reproduction, things like that. But if we have all of our information available to us, that's essentially the library. Whereas then when we go down into the DNA itself, if we remember back to high school and the Dewey Decimal System, and they're organized by categories, one shelf can be that chromosome, which is where our genetic variation lies and where our DNA is organized. We can then break that further into books and paragraphs, where that book is then a gene, the DNA sequences themselves are the sentences, and as we go further down, that alphabet essentially is our DNA base pairs. So those are, I like to joke with people that, if I make a typo or a spelling mistake, that, I'm a geneticist, I only deal with 4 letters at a time, sometimes 5 with RNA. So, try to have some grace for me, since I just deal with those 4 letters most of the time. And, one benefit for the sheep industry is that we do have a sheep genome available to us. So this is research that's been done, and essentially the same thing's been done with the human genome. You may have seen in the news that recently the human genome finally has a telomere to telomere, which is just saying that they have a more complete genome sequence. We aren't as complete on the sheep side, but it has allowed us to map certain traits and certain genes to certain chromosomes. So here we have our 1 through 26, our X chromosome as well. And then there's some other information here, but really the important thing to take away from this graph is, we know where myostatin is, we know where the agouti signaling protein is for coat color, we know where a large number of genes that play a role in our economic traits, we know where those are on the genome. So that allows us to then have more direct and applied research in figuring out marker panels people can use for selection, or what changes in the DNA cause differences we see in our economic traits. And sheep have 54 chromosomes, so we've got 27 pairs, so that's 26 plus our sex chromosomes. We can compare that to our other species. We're kind of right in the middle, so humans have 23, dogs have 39. Really, it's about the same number of DNA base pairs, and just organized a little bit differently. In the sheep genome, there's about 30,000 genes. It's been estimated, and again, that gene is just that region of DNA which is going to code for some functional end product. And like I said before, genes are located on chromosomes. There's some other terminology here. Locus is just saying that physical location. When we're talking about our important traits, we often refer to areas that are associated with that trait as quantitative trait loci, or QTLs. So that's just saying that that section of the genome is associated with variation in our phenotype of interest. And it's important to remember that every sheep has 2 copies of each chromosome, so they're diploid. So that's just saying that there's 2, and the ploid is just referring to those chromosomes there. So just like sheep, or just like humans, we inherit one from the paternal side and one from the maternal side, and both copies of these chromosomes may not be identical, which is what leads to some of our variation, and that then leads to having different alleles for each of these genes. So we can have homozygous or heterozygous allele pairs. So homozygous is just saying that they're the same, like we can see in the top here where we have the 2 capital A's. It could also be 2 little a's for just kind of how we're designating those alleles. Or heterozygous is just saying that those 2 alleles are different. And there's a number of different mechanisms for how these alleles interact with each other, which one may be dominant, or if they're going to be contributing additively, so combining the effects of both of them. It really depends on the trait in terms of how they're going to interact with one another, but that's what people like me try to figure out, so that you guys don't need to do that type of work. And we're diploid because of the way reproduction works in mammals. So the sperm and egg each contain only 1 copy of this, and then they then form that diploid during fertilization. And again, they might carry those different alleles. So to get us started off, there's a number of simple traits where this allele notation can be really helpful. So if you're a small flock producer, or not even small flock, but say you're breeding for coat color, since you have a hand spinning flock, these simple traits oftentimes are controlled really only by 1 gene, which makes it fairly easy for us to utilize genetics in terms of selecting for the trait that we want. This also applies to the Scrapie susceptibility codons, as well as a number of others such as spider lamb, myostatin, and then there are more out there. And these are oftentimes now being incorporated onto some of our marker panels or SNP chips, so that you don't necessarily have to pay for an individual test. But the beauty of these simple traits is it's relatively straightforward for once you have a number of your animals genotyped, so you know what that DNA sequence says they should be, you can then use that moving forward to predict what those offspring are going to be like. So because I grew up with Romneys and have a pet projects in coat color, we'll just kind of run through this example of using this brown locus. So this determines whether your sheep is black or brown. We're not going to talk about how white overrules this, but in this case, there are two allele options, either black or brown. Black oftentimes is denoted with the plus, and brown is with this little b. We know it's in this TYRP1 gene, and oftentimes genes you'll just see reduced to an acronym, makes it a lot easier rather than spelling out, tyrosine ACE related protein 1. And we know that there are 3 molecular variants. So there's 3 changes in the DNA that cause these animals to have brown wool as opposed to the black wool. And it is a recessive inheritance pattern. So what this means for us, if I can get my slides to work, is we can use what's called a Punnett square to determine what those offspring are going to end up as. So here we have the sire on top, and say we know his genotype is black and brown, so that plus and plus sign, excuse me, and the little b, and we know the dam has the same genotype. So what we can do with these Punnett squares, and this applies to all of our simple traits where we know the alleles are genotyped and such. And we can apply this to oftentimes Scrapie codon. If if you have individuals that are RR, you want to breed with QQ or QR and determine the frequency. So how this works is, you then split your 2 alleles because each of these is residing on a separate copy of the chromosomes. And then you can just trace, again, if I can get my slides to work here, you then trace those alleles out to each box. So this first black allele from the sire gets put into these 2 boxes here. We then have the other allele, the brown allele in our sire, go to the 2 boxes on the right. We then do the same thing for the dam, for both of her alleles that she's contributing. And this gives us then a frequency of how likely or what the proportion of the offspring genotype should be. So in this instance, we end up with a homozygous plus, homozygous little b, and then 2 heterozygous individuals in either diagonal. So what we can then translate this to is, 25% of our offspring should be black based, and having the homozygous plus, half of them should be black, but be carriers for brown because black is dominant to brown. And then 25% will be the little b, and should be phenotypically brown. But like I said before, this can be applied to any of our simple traits, Scrapie codon susceptibility being probably the one most applicable to the most of you. But what about our other traits? Again, if I can get my slides to work. Not all of our traits are simple. Unfortunately, really, most of the traits that we care about and that are going to be paying most of our bills are complex traits. And what that means is that there's more than one gene involved and there's a much larger environmental influence. So what that most often means is, there's a larger role of environment on these traits. So an example of this is, if we feed our sheep more, generally that's going to be leading to a higher weaning weight. So we do have to factor those into when we're evaluating these traits. So, these types of traits include our weight data, so weaning weights, adult weights, reproductive performance, and carcass traits oftentimes. These also can include your health traits, so immunity or fecal egg count. Really, most of our traits that we haven't tracked down to just a single gene or 2 genes involved with really do fall under this complex trait and is really where most of our bills are going to be paying for. So how can we work with these traits since we can't make Punnett squares, or we can, but it's much more difficult to make Punnett squares with 10 genes involved, or 100 or 1,000 or however many there are. So really what we need to do is we need to select. And really this is just our process where we're making certain traits more prevalent, really good example of this is the Jacob spotting. So, during that breed development, they selected just for those spots and for certain characteristics. So that became fixed within that breed. But we can select on really any trait at all. We can, oftentimes we're selecting for increased weaning weights. We're trying to select for other traits that may not result in an direct economic benefit, but things that will make our lives easier, so lower dystocia. But really, any trait at all that we can measure, we can do selection on. So how do we go about doing this? There have been, I'll say, historically, some not so well designed ideas on this. So one prime example of this is, we can select for increased body folds to increased wool production. And while that did increase wool production, there was the unfortunate drawback of increased prevalence of fly stripe. So this is predominant mostly in merinos, and down in Australia, they perform the selection. And there are a lot of difficulties with selection. Unfortunately, oftentimes we're not producing just one product. So we do have to strike that balance between meat, wool, and milk. And some of these traits, like I said, they're complex, but they're also hard to measure. So, out of season breeding or out of season lambing success can be difficult. Immunity can be difficult. And it can be a challenge to select on something that is hard to measure, or it takes a very long time to get any phenotype information on. But there are ways, to kind of counteract, or there are things in our favor that we can use to our advantage to determine, how fast we're going to go or how accurate we're going to be in our selection schemes. And the one thing that plays a role in this is how heritable our traits are. So all that's saying is how much of the trait is due to additive genetics in this case. So oftentimes we can categorize them into our, broader categories of repro, growth, etc. And for the most part, our reproduction traits are oftentimes our least heritable. So there's a lot more environmental noise in these traits. So it can be harder to select for, or it's going to be take a much longer period of time to see the progress we may want to see in a particular trait. As we work our way across the slide here, growth traits tend to be low to moderately heritable, similar for our carcass traits. So this is essentially what these numbers are saying here is, about 15% of the variation we see in birth weight can be attributed to our additive genetics. Whereas fleece traits tend to be the most heritable, so we can make relatively quick progress on the fleece side of things because there's a much larger genetic component to those traits versus, say, fertility of 0.05, and we have a almost greater than 10% or 10 times the amount for staple length there. So we can use this to our advantage. So we know that our fleece traits are going to take a little less time to get where we want, whereas our repro and growth traits might take a little bit more time. So what we can do to try and balance this is put a little bit more emphasis on selecting for some of that growth and repro, because we can go back and fix the fleece traits relatively quickly. And now, get onto a soapbox here, and I'm sure it's been hammered home in other genetics talks you may have listened to, but you can't manage what you can't measure. So in order for you to do any form of selection and be successful, in my opinion, you need some type of identification, whether that's ear tags, tattoos, paint brands, or more commonly now, or becoming more popular, is electronic ID tags. So these EIDs, you can make better progress in terms of overall genetic merit of your animals if you have individual identification. You really can't, you can't manage what you're not measuring. If you want to improve a trait, you need to put some type of metric on it, and it can be subjective. It doesn't have to be a decimal point number for your evaluation. It can be as simple as, I'm just going to give a number to, okay, this lamb has a lot of wool cover on its face and this one doesn't. It can be as simple as that if you want to make progress on that. But you have to do some type of measurement on it if you want to improve it. So what are some of the tools currently available to shepherds for And I'll say this is more focused on kind of a mix of molecular and more traditional quantitative genetics. But the first one that's available to you is parentage panels. And I'll say these tools are available outside just general data recording and flock recording within your own flock. But these are kind of extra things that you can look into to see if it would be economical for you to utilize. So the first one of these is parentage panels. So this oftentimes requires some type of biological sample, whether it's blood, you can extract DNA from the bulbs of hair, tissue, fluids, etc. We can get DNA from that on our animals. We can then use that DNA to determine our parentage. And there's a number of options out there for you. I'll just put in here now, since this is also being recorded, this is not an endorsement of any of these operations or services. This is just listing what your options are. So if you're using a multi-sire mating groups to reduce some of your labor costs in the fall or spring, whenever you're breeding for, parentage can be an extremely useful tool, especially if you're going to be using, say, the National Sheep Improvement Program, or trying to get a better sense of who sired, how many lambs, and whether they're actually doing their job. And it's also, hand recorded pedigrees. It's estimated across livestock industries that there's probably about 10% error rate, which as a scientist is a little more than we would like, but that's, unfortunate side effect of, human nature. We make mistakes and those get recorded and passed on. And really with DNA is we're able to correct for that. So there's a number of options available. So those are listed there, and then the technology varies as well as the price. But what this allows us to do is, initially say we put two sires in a breeding group. We know who the dam is because we saw the lamb come out of it, but we don't know who the sire is. So what the genotyping allows us to do is we can then say, and this is a really simple example of just genotyping two spots, but oftentimes we're genotyping, 100 or more nucleotides on these animals. We can then say for certain that, okay, if our dam is capital A and capital B homozygous, and we know the sire one is capital A homozygous, little b homozygous, and the other is little a homozygous and little b homozygous. If we genotype our offspring and they're, end up with this, so it's heterozygous, the little a, and homozygous, the big B, really we can rule out sire one as an option, because there's no way, with the genotype of the dam, that little a could have come from sire one. It had to have come from sire two. And similarly, since there is no little b allele in our offspring, and it's just the large B, we can also rule out sire one based on that. And that's oftentimes how our parentage panels are working, is they're excluding potential sires. But then this does allow us to have a much more accurate pedigree and make much more accurate selections, since we can attribute those offspring records back to a particular sire. And there's pros and cons to these other these different companies, as well as price and the technology. It just depends what you're looking for in terms of some other things available. There's also single gene marker panels or marker tests. These are becoming less common as some of the more extensive panels are developed. And oftentimes, the most common one of these is testing for Scrapie susceptibility, and our top three codons, 171, 154, and 136. Again, a number of different companies offer this. This is generally a $10 to $15 test if you're just doing one of these. So it might make more sense to use a panel that's been developed that contains a larger number of SNPs or markers. It might cost a little bit more, but you're getting more bang for your buck with those. And similarly, like we saw before with our Punnett square, we now have done this now for, we've put two genes or two codons for the Scrapie susceptibility here. So the A, V is from the 136 and the 171 is the Q and R. And we can then do the same thing as we did on earlier and follow these out to each of these squares to give us an idea of, okay, if we have a ram that passes on the AR and the U is say VQ, what is that offspring susceptibility going to be like? Or if we've got an AAQQ animal breeded to another animal of the same genotype, how susceptible is that animal going to be? So this is most commonly used in our single gene tests. There are also a number of other ones available, such as spider lamb, most commonly in Suffolk, or the hairy lamb, ectodermal dysplasia, often in South Downs, but others are available. Calpege, myostatin, and OPP susceptibility are some of the other single gene tests that are commonly offered. There might be some others that are more recent, but these are likely the most common ones. The last tool I'll spend a bit more time on is the National Sheep Industry that's a granting agency, National Sheep Improvement Program. And what this is doing is except for the Katahdins, it's still taking a quantitative traditional approach to estimating breeding values. So in a nutshell, what this is doing is combining pedigree information, which is why those parentage panels are extremely important, or it's really important to know who the sire and dam are. It's combining that with our phenotype information, whether that's growth rate, fiber quality, etc. It's then applying, a mixed model equation to estimate the genetic merit of that animal or all the animals within your flock. So then the goal is, based on that, we can breed for superior animals because of that predicted genetic merit. So how this works more logistically is, as a breeder, you submit phenotype and pedigree information to LambPlan, which is run by Sheep Genetics in Australia. That gets run twice a week, and that's generating our breeding values. So the NSIP members, we pay membership and data fees to NSIP. They provide tech support and service to the members. We ship data to LambPlan. They send us EBVs back. And then NSIP works with LambPlan both on the data fee side, as well as developing new technologies. So NSIP's technical committee worked fairly closely with LambPlan in implementing the genomic enhanced breeding values for the Katahdin breed, as well as any other additional improvements that are going to be made in the future. That's worked in conjunction between the two. So how does this all work, by itself, outside of NSIP? Excuse me. So if you're going to be, kind of the back end of NSIP, should I say, how this is working. So really what this is doing is, say we have two animals here, and the best school colors that you can find for MSU here, we've got our two sheep, and we have the question of which animal is best. Now we can measure our trait. So in this instance, we'll just say we're using fleece weight as our example trait, and we want to know which is best. We can pull in information from our pedigree. So we measured it on our individuals. So the blue one here had a 10-pound fleece weight, the yellow had a 13. Now we pull in our parentage information that we have accurate, and we can see, the blue parents had a higher fleece weights than the yellow parents. But NSIP goes broader than that. So it's not just pulling in from the parents, it's pulling in from the grandparents, it's pulling in from siblings, half siblings. We have all this data combined, and we can plug that into an equation that, if you want to go more into it, I'm happy to talk more about it, but just for simplicity's sake, we plug it into our mixed model equation, and we're able to then generate our estimated breeding values. So even though when we look at our individual, just the raw phenotype, our yellow individual here had a 13-pound fleece weight, when we factor everything in from all the relatives' performance, all the parents, all the grandparents, et cetera, all the relation performance, we actually see that the breeding value for our blue animal is higher. So what we see in our phenotype may not be the animal that is going to produce the better generation or better next generation. Oftentimes, there's a lot more environmental influence. So, hey, maybe this guy outcompeted everybody at the feed bunk. So he just got a lot more feed, so he had a higher fleece weight, or something of that nature, where it caused some of this environmental noise that NSIP is able to remove a good majority of it to allow us to say, okay, well, really, this guy, this blue animal is going to be producing more fleece or pounds of fleece, pounds of wool than the individual that looked better initially. So NSIP does have a large number of tools available. They do it for a large number of traits. So here, depending on our age category, our measurement types, or our weaning weights here, what their breeding value is, and their accuracy. So the ram buying guide is a really good resource for folks wanting to learn more about interpreting these. And really what this is for this individual, our breeding value is 3 kilograms. So this is saying that ram, compared to his contemporaries or those individuals he was raised with, he has the potential to be 3 kilograms heavier. And then because the sheep industry is a bit different than cattle, we use the breeding values. If we wanted to see then what our progeny differences are, so for beef cattle, those are our EPDs, or expected progeny differences. For dairy cattle, it's parent transmitting ability. We just essentially half that number. So instead of 3, it'd be 1.5. We would expect that animal's offspring to be 1.5 kilograms heavier due to the genetics of that sire. So these can be really useful depending what we're looking for, or what you are looking to improve, and whether that's growth rates, fleece traits, carcass traits. There are a number of metrics available through NSIP to allow you to do that. Now, we don't want to select just for one trait because that can lead to problems. So we do try to have some balance across our traits so that we don't lose progress in any one trait, and we make progress kind of across all of them, hopefully in the most economic way. So that's where these selection indexes come in. So this is trying to rank individuals by their overall economic merit. And this is really the most efficient way to select for multiple traits. There are a number of things that we do require for this. So we do need to know the economic importance of each trait and how much a unit change in that is going to affect. So, seeing a pound increase in weaning weight or sale weight is going to bring more economic value than seeing the pound increase in fleece weight, because we have a better price for our land. Well, maybe not at the moment, but traditionally most of our revenue is going to be coming from the lamb side. We also need to know how much genetic impact or control of that trait is available in the heritability, as well as then correlations between those traits with an index, and then we can put it all together. So we have a single number or a single value to select from, rather than trying to do this on a one-by-one basis. So for an example of this, and what this looks like, is this Western Range Index. And this is shown as a value. We start with a base of 100, but then we add and subtract values depending on the importance or how economically important it is for the profitability. So the range index was developed mostly for Targees, but also applied to other breeds such as the Rambouillets. And we can see in here, so we start with our base of 100, then for every unit increase in our post-weaning weight value, we see a 2.2 increase in our index. We see this similarly for our maternal weaning weight. So this is the maternal contribution. But then we do have a slight negative impact on yearling weight to try and keep that mature size down so we don't have to feed as much. A slight increase on fleece weight, a slight decrease on fiber diameter, so we select for finer fleeces, and then a heavy emphasis on number of lambs born to increase overall production. So this has been relatively successful over the past, I guess, 2 decades or so since it's been around. And this is a way to kind of select for multiple traits. As long as we have some economic information, we can tie it all together. And then by selecting just on this number, we should be making progress across all these traits in the most economical way possible. So really, to start to kind of wrap up, really take-home message from this talk is selection can work on any trait. We just have to measure it. And it might take a little bit longer depending on how heritable that trait is, or it might go pretty quick. But it can work on any trait that you want to measure. It really just depends on what traits are bringing you money. And those are the ones you should focus on for selection. That's not to say you should ignore things like conformation or other factors, since you still have to like what you see when you look out in the pasture. But really, it should be focused on those traits that are going to bring you money, and that's going to vary by flock to flock. Second, you can't improve what you can't measure. I am a strong proponent of ear tags or electronic ID tags or some type of identification for your animals so that you can get individual data on an individual basis for selection. That makes ideally your life easier by then making better genetic progress or more quicker but genetic progress. It also makes my life easier if I ever want to work with you, because it makes research go a lot smoother if you have IDs linked up with individual data. And then finally, there are a number of genetic and genomic tools available, whether it's parentage marker panels, NSIP. NSIP is going into the genomic field as well, with testing on a 50,000 SNP marker panel. But the genomic enhanced breeding values are just in the Katahdin for the moment, so I didn't spend too much time on it since we're in the Intermountain West here. But then there are plans to bring up some of the other breeds on genomically enhanced breeding values as well in the near future. So there are a number of tools available for you to make genetic progress there. And I'll just kind of put an aside here too. We didn't really talk about inbreeding or crossbreeding, but those are tools available to you as well. And with that, I'll open it up, take any questions. I've also got my contact info there. Feel free to send me an email. You can find me on the web, so you can call as well, but I'm happy to answer any questions you may have.
Awesome. Thank you so much. I'll remind all of our attendees that if you would like to ask a question, go ahead and shoot it into the Q&A box, and we'll make sure that we talk about that. I'm going to kick it off with a couple of questions for you. The first one that I was thinking about is if somebody were wanting to go into working with the NSIP program, what would be kind of the cost benefit of utilizing rams that are having those EBVs? Because we all know we have to pay a little more for them. What are we going to get out of that?
Really, the biggest thing you get out of that is more confidence in what those offspring are going to be. So yes, you can go to the sale barn or go to your neighbor and buy a ram that doesn't have NSIP numbers for $400, $500, or whatever their going rate is. Your NSIP ram, yes, you might pay $1,000 or upwards of $1,000 for it. But if you have confidence that that ram is going to be producing heavier weaning weights, so say that weaning weight breeding value is a 3 or 4, so the offspring should be 1 or 2 pounds heavier. If you're a commercial operator and you're running, say, 1,000 ewes, really all your lambs should, in theory, be 1 or 2 pounds heavier at weaning, which, if you do the math, should more than pay for that ram after one breeding season. And then, if you use him the next year or the year after as well, you do have a fairly quick turnaround in getting that money back. It is an investment up front, but there you have a much greater confidence in that ram's genetic potential to pass on. It is scary. I'll say the first time I bought a ram that was $1,000, I was not happy, but he worked out quite well for us because he had those NSIP numbers.
Right. So what's your recommendation then if you did go ahead and jump on board and buy that $1,000 ram or whatever it may be? From a ewe management perspective, what would be some tips that you can give people that maybe would allow them to really benefit from that genetic progress?
So talking about his daughters then
Yeah.
now, or okay. Yeah, so we have a number of other traits. So it's not just the carcass traits, but if you're going to keep ewes back, and they say they're the commercial ewes, but that ram, then you have information on his genetic potential for, say, number of lambs born, number of lambs weaned, those maternal weaning weights and things like that. You can pull all that in, and those daughters that he produces, you should have a better sense of how they're going to perform because you have those numbers on him. And so you can then see if, say, you want to increase your prolificacy or number of lambs born in your flock, you can use a ram with a higher number of lambs born estimated breeding value And that should then in turn produce daughters that are more prolific. So, because we can't measure lambing rate directly on a ram, since they don't, do that, we can pull in the information from all of his female relatives that have been bred and such, and that gives us a much better sense of, okay, yes, this ram will increase the prolificacy because he's got, say, I don't know, an LB of 0.30 or 30% more than his contemporaries. So it definitely can be used on the use side. And similarly for the weights, really any of the traits you want to select on, if you want to improve some of that maternal base as well.
Great. So if you could recommend one tool to somebody who's never really worked on any genetic progress in their flock,
yeah,
which one do you think you would have people start with?
So it's not necessarily a direct genetic tool, but probably the most economic tool for somebody to get started off with is ID system, whether it's just regular ear tags printed that you get at Murdoch's, Tractor Supply, wherever, or you go with electronic ID tags from Allflex or Shearwell or one of those companies. Just some type of measurement or a way to put individual measurements to an individual ID. I think that's, if you're not quite ready to make that jump into NSIP and joining an NSIP, I think electronic ID tags or ID tags in general, if you don't have any system in place, are the next step for you. It just allows you then to say, rather than looking at the group and saying, okay, this group of 300 ewes had an average lambing rate, we ended up with 1.3. You can say for sure, well, okay, this ewe didn't have a lamb this year, she goes down the road. So it really is allowing you to cut out your bottom end. And that's really what a lot of these tools are doing, is cutting out that bottom end, not necessarily getting that top all the time. But definitely ear tags would be, or some type of ID would be my first investment.
Right. And I mean, that comes into play for a lot of different things, including, biosecurity and nutrition and all of that. So I agree with you. We had a comment from Tom Perkins about the percentage of accuracy, and I'm wondering if you can speak to kind of what the cutoff should be if you're looking at those accuracies as far as purchasing.
Yeah, so it's going to depend on the trait. So with the Rambine guide there, it had the accuracy of 94%. Oftentimes you don't reach that accuracy until you have hundreds of lambs on the ground. So as NSIP gets more data, you'll have a higher accuracy on that trait. Some traits will be relatively low accuracy, just given the nature of the low data. So some of our fertility measures generally are lower. What that accuracy is telling you is, if you imagine a bell curve around that number, or a normal distribution around that number, the higher accuracy, the narrower that curve is. So the more likely we are to that true breeding value. So if your accuracy is 10%, your number's 3, or your breeding value is 3, you might end up being down around 1 or 2 or 4 on either side of that number, just because of that lower accuracy. If you go through onto the NSIP's website, they've got a searchable database that lists some of the accuracies for the traits as well. Really, it depends on the breed. So like the Targhee breed, our accuracies for our fleece traits and our weight traits are relatively high. So they're anywhere from 60 to 90 and up for those two trait types. Is oftentimes just because we have a lot of historical data. I think there's upwards of 10,000 records since NSIP's inception. If you're working with a different breed that has fewer records, it might be lower. So I don't really want to give a threshold for what accuracy to use, because it's going to depend on what you're most comfortable with in terms of your risk, as well as the breed. And I'll say even an estimated breeding value with an accuracy of 10 is better than just using the raw phenotype, in my opinion, because it's just better because you're taking in more data.
Right. Yeah. So along those same lines, we have a comment from Tom Boyer about participating in ram sales and having the need for 50 rams or whatever it may be, and they're not available. So maybe we could talk about how we as an industry move forward with providing better opportunity, and maybe, if there's ways that people can get involved to improve some of this, or what do you think on that topic?
Yeah, that's a good comment, Tom. Unfortunately, I couldn't go there this year. But yeah, that is tough, to meet those large orders. And I think one thing that could be a potential opportunity is maybe those individuals that, they may not be coming direct from an NSIP flock, but kind of that next step down of a multiplier flock. So say that flock is using NSIP rams and maybe doesn't have their U base on NSIP, but then the offspring of those should, if we are making genetic progress the correct direction in our traits, those offspring should be better in theory than industry average. So that might be a way to fill some of those larger orders. I like to be of the view that a rising tide is lifting all boats. So it may not be sales directly to you, but you're encouraging more people to raise sheep and more people to buy your rams for that kind of multiplier flock before it goes on to kind of that commercial flock. I think oftentimes we're skipping that step, which isn't a bad thing per se, but then it does become difficult for us to have data on all of the rams that are going to be used in commercial operations, or have rams available with data for those commercial operations. So I think if there's a way to kind of get that step in, that might be a way to meet some of those larger orders. Is it a perfect world? No. Or a perfect solution? No. Because ideally, yeah, I would have every sheep on NSIP if I could. But I think that could be one way to try and meet that demand, since it's better than nothing. But it is either until you have more producers enroll in NSIP, I think that's kind of a good stopgap, until more folks get on NSIP if they're going to be raising those rams. Yeah, I don't have a good answer for you on that one, Tom. If I could clone, 50 best rams or start a cloning operation and just pump them out for you, I would, but that'll cost a lot more money than I have in my startup.
Yeah, exactly. That's the challenge, right? Money.
Yeah.
But I think, as we look into the future, we're seeing a lot of our industry representatives moving this direction. And I think there's a good chance that it's going to continue to get bigger as we go forward. So
yeah, absolutely.
As always, we're a tiny bit behind the beef industry, but they're in that boat now. So we will be eventually, right?
Yeah. And we can learn from their mistakes.
Exactly. So, all right. Well, I think that kind of wraps up the questions that I have for you. And I just want to thank you again for joining us today. And we'll have you back again at some point, because we all like to hear all the different topics that we have available to us. So thank you all for attending today. And again, if you don't mind filling out the survey as you get off the webinar, that would be great. And we will see you next month.